Ribosomes. Ribosomes are very much essential cellular organelle for each kind of cell because ribosomes play a crucial role in protein synthesis. That means it binds the amino acid together and makes a polypeptide chain, and that ultimately leads to a construction of a protein. But if a cell lacks of ribosomes, the cell cannot survive because it cannot make up any kind of protein. So ribosome is very much essential for a particular kind of cell, in case it is prokaryote or it is eukaryotes. Hello friends, welcome to Biology Concern. I am Animesh, and in this video I am going to discuss about the ribosomes in prokaryote and eukaryote in a brief manner and each of the aspects covering the ribosomes. So you will understand how ribosomes produced and the main functions of ribosomes and how it differs from prokaryote to eukaryote. So let's jump into the topic. So ribosomes basically are ribonucleoprotein. So what is called about ribonucleoprotein? Ribonucleoprotein, that stands for, it consists or made up of a RNA and protein. So it is made up of RNA as well as protein, so it is called as ribonucleoproteins. Next one, the ribosome consists of two unequal subunits. One is large subunit and another one is a small subunit. Both the subunits, that is a large subunit and small subunit, both of the subunits make up a complete ribosome. Okay. So ribosome, the functional ribosome consists of two different subunits of unequal size. One is large and another one is small. And ribosome have three different sites inside of it. One is known as A site, another one is known as P site, and third one is known as E site. So one is known as A site, another one is known as P site, and third one is known as E site. A site stands for amino acyl site, where amino acylation of tRNA is taking place, and second one is the P site, that is the peptidal site, where two amino acid form the peptide bond, and third one is the exit site, from where tRNA leaves from the ribosomes. Okay, next thing is that the main thing is that when it is a protein, ribosome is actually a ribonucleoprotein, so it consists of RNA and protein, right? So where from it begin or where from it synthesize. So if we consider this is a cell and this is the plasma membrane, this is the plasma membrane, this is the nuclear membrane, and this is the nucleolus. So main gene for ribosome, that is the rRNA genes that make up of those kind of ribosomes, are resides inside of the nucleolus of each and every gene, each and every DNA. In eukaryotes, the large subunit make up of three different kinds of rRNA and one protein. That means it consists of 28S rRNA, 5.8S rRNA, 5S rRNA, and 49 proteins. So 49 proteins along with those three kinds of RNAs makes a ribonucleoprotein subunit, that is a large subunit of eukaryotic genome. And here 18S rRNA present inside of small subunit of ribosomes. Okay, so here 18S, 5S, and 5.8S, those three RNAs are make up a cluster of ribosomal genes, that is the rRNA genes, and produced inside of a rDNA region. Okay, here 18S, 5.8S, and 28S, that is the rRNA genes. Those rRNA genes are organized in a single transcription unit and transcribed by the RNA polymerase 1. So those three genes are reside or make up a single transcription unit. That means when RNA polymerase 1 moves along with those DNA, rDNA, they actually make up of 18S, 5.8S, and 28S rRNA genes all together. And in order to produce large amount of RNAs, the transcription units encoding the RNA are present in multiple copies in tandem repeats. That means those copies, here one is 1 copy, another is 2 copy, here is a 3 copy and 4 copy. So these genes are in a tandem repeats. Okay, tandem repeats means these genes are occurred one after one, one after one, one after one, or these genes resides one after one. So these genes are stayed in a cluster and they make ones after a one, and these genes are resides one after one, just one after one, one after one, one after one, and that's why it makes up a tandem repeats and it makes up a rRNA gene cluster. And another one is 5S rRNA. So 5S rRNA, those 5S rRNA is actually transcribed by RNA polymerase 3. So the 5S rRNA is actually another region of present in another region of the particular rDNA, and it is transcribed by RNA polymerase 3, whereas 18S, 5.8S and 20S rRNA is organized in a single transcription unit and transcribed by RNA polymerase 1. So that is the main difference between two of the rRNA genes. That is the 5.8 rRNA and 18S, 5.8S and 28S RNA. All three together make up a cluster of gene, and those gene resides in a tandem repeats, that is one after one, one after one. That is the resides in this kind of manner. Okay. So in humans, in human chromosomes, chromosome number 13, chromosome number 14, chromosome number 15, chromosome number 21 and chromosome number 22 contains rRNA genes in a region called nucleolar organizer region. So here in case of us, in case of human, the chromosome 13, 14, 15, 21 and 22 contains those rRNA genes in a region called nucleolar organizer region. So where in the nucleolus, those kinds of in chromosomes, those kinds of genes or rRNA genes are present with inside of a nucleolar organizer region, and that is the most important region which holds the all rRNA genes together. Okay, so if we look a causal look of prokaryote and eukaryote ribosomes, then we observe that prokaryote have 70S ribosomes, whereas eukaryotes have 80S ribosomes. So what is mean by 70S and 80S? When small subunit along with the large subunit, both subunits are together, they form those 70S subunits, and in case of eukaryotes, those have the 80S subunits. So what is mean by A's? Okay, so in case here in prokaryotes, the 70S subunits further divide into 50S plus 30S, 50S plus 30S, and in the eukaryotes it is divided into 60S plus 40S subunits. So when I'm a student, naturally there's one question came up in my mind, that 50 plus 30 is equals to 80, right? And 60 plus 40 is equals to 100, right? So why it is called as 50 plus 30 is equals to 70 and 60 plus 40 is equals to 80? So that one question came up in my mind, and ultimately I find the answer when I studied biophysical instrumentation technique, that is known as centrifugation technique. So S here present in the S stands for sedimentation coefficient. So here is a machine called as centrifuge, and in the top view there is a chamber like that, and it actually a rotor. And it spins in very fast, and when we put our centrifuge tube inside of this particular machine, those centrifuge machine, those machines are separated those kinds of cellular organelles, RNAs, DNAs and proteins all differently. Okay, so in various time, in various aspects of time, they actually, those machine or centrifugation technique helps us to separate RNA, DNA, cellular organelle as well as the particular proteins from a cell. So this is a technique from where those kinds of those subunits are separated. When large and small subunit binds together, they separated in 70S time, and when they are individual in nature, they separated in 50S and 30S time. So S stands for sedimentation coefficient. So S stands for sedimentation coefficient, and one is the unit is known as Bedburg unit. So unit is known as Wedberg unit, and 1s stands for 1 into 10 to the power minus 13 seconds. So the simple calculation is that when we talk about the 70s in nature, so we talk about 70 into 1 into 10 to the power minus 13 seconds. So in that time, 70 into 1 into 10 to the power minus 13 second, in that time, those large and small subunits precipitates together within a particular centrifuge tube. Okay, and when they are single, they are dissociate from one another, that is the large and small subunits. Each of the subunits precipitate their own time. That means 50 into 1 into 10 to the power minus 13 second, and 30 into 1 into 10 to the power minus 13 second. Okay, if we take a closer look of 50s and 30s along with 60s and 40s, we observed that those subunits are actually made up of further subunits, that is make up of 23s rRNA, 5s rRNA and 34 proteins in case of 50s subunit. And the molecular weight of the 50s subunit is 16 lakh Dalton. In case of prokaryotes, in case of eukaryotes, it is 28 lakhs Dalton, and it is made up of 28s rRNA, 5.8s rRNA, 5s rRNA and 49 proteins. In case of small subunits, the molecular weight in case of prokaryote is 9 lakh Dalton, in case of eukaryote it is at 14 lakh Dalton, and here in case of prokaryotes we found 16s rRNA along with 21 proteins, and in case of eukaryote we found 18s rRNA along with 38 proteins. In case here 23s rRNA, they consist about 2900 nucleotides. In case of 5s RNA, they are consist of 120 nucleotides in them. So basically, what is the sedimentation coefficient and what is about those particular things? Sedimentation coefficient is the rate at which sedimentation occurs in the centrifugation is expressed in terms of sedimentation coefficient. That means when a particular molecule, when a particular cellular organelle precipitated in the bottom of the centrifuge tube, it is known as sedimentation coefficient. And sedimentation coefficient is formulated by S is equals to V by omega square R. V by omega square R, where V is equal known as sedimentation of the particular molecule, omega is the rotation of the rotor in radian per seconds. So the rotation of the rotor in rad per second, and another one is R, that is the distance from the central axis of and the particular centrifuge tube. Okay, so from central axis to particular centrifuge tube, this is the R. Okay, so those kinds of things are actually makes those sedimentation coefficient or equation of sedimentation coefficient. So this is how we talk about the prokaryote and eukaryote ribosomes, and now we clearly understand that 70 is not a mathematical calculation, but it is considered of a centrifugation technique and sedimentation coefficient. So 50s subunit, that means when a large subunit precipitate in which time, this is known as 50s, and when a small subunit precipitate, that is known as 30s. That means 30 into 1 into 10 to the power minus 13 seconds, and in case of 50s, 50 into 1 into 10 to the power minus 13 seconds. So altogether, when large and small subunit came up together or binds together, they actually precipitate in that time 70 into 1 into 10 to the power minus 13 seconds. So this is about all about prokaryote ribosomes and eukaryote ribosomes. I hope this video is helpful for you to understand about 70s, 80s, and what is about 60s, 40s, 50s, 30s, and what is the rDNA, what is the gene cluster of particular rRNA genes, and what is the nuclear organizer region. So I hope this video is really helpful for you. If you like that video, please hit the like button. Don't forget to subscribe, and thanks for watching.